are a way to test new methods of diagnosing, treating, or preventing health conditions. The goal is to determine whether something is both safe and effective. A variety of things are evaluated through clinical trials, including medications, medication combinations, new uses for existing medications, and medical devices. Before doing a clinical trial, investigators conduct preclinical research using human cell cultures or animal models. For example, they might test whether a new medication is toxic to a small sample of human cells in a laboratory. If the
Yes, Sharon, this much is fine.
okay.
Now let us understand. You can see in the first para, it is written clinical trial are a way to test new method of diagnosing, treating, or preventing health condition. So here they are telling what are or what is clinical trial. So it's a way to test new methods to diagnose, to treat, or to prevent health condition. The goal is to determine whether something is both safe and effective. It means the ultimate goal is to create a drug, or if you are creating an medical device, so it should be safe and effective. Then a variety of things are evaluated through clinical trial, including medication. So during the clinical trial, or what is the purpose you do clinical trial? What For what reason you are doing clinical trial? So obvious, to create a medication, either to create a medication with combination. Sometimes, like doctor give you combination of drug that take these three drugs together in the evening, morning, and uh, whatever time they suggest. Okay. So, and then new uses of existing medication. Sometimes the clinical trial happened to figure out the new uses for existing medication. And the next one is medical devices. So all the devices which is used in the hospital, for them also clinical trials are conducted. Okay, it is not like directly they have purchased it from the market. So it should be clinical trial proved, or it should be clinically certified. So majorly the clinical trials are done for the medication actually and medical devices. The end goal is to create a drug which should be safe and effective or treat the disease, or a medical devices. Now, before doing a clinical trial, before starting a clinical trial, investigators. Now, who are in this investigators, guys? They are, or they can be doctors.
Doctors.
Correct. Doctors or a team of experts, clinical experts. They conduct preclinical research using human cell cultures or animal models. In the past, you might have heard this, like a lot of NGOs, especially animal NGOs, they oppose this, don't test drugs or things on animals. Because what's the common practice of the industry, that whatever they are going to create, they first gonna test on animals. Okay, like rats or monkeys. Okay, so what they're telling, before starting the clinical trial, a preclinical research will be done on human cells and animal models. For example, they might test whether a new medication is toxic to a small sample of human cells in laboratory. So why this preclinical research is done? To ensure before testing on humans, the drug is safe. They test on animals and human cells. If the preclinical research is promising, they move forward with a clinical trial to see how well it works in humans. So if the preclinical research seems to be proven and safe, the main purpose is it should be safe. So then they'll move to the humans, because they cannot directly test on humans. because till now it is considered as humans are more important than animals, and that's how it goes. That's why they test on animals first. Okay. So the clinical trials start from now. After preclinical research, if the drug is safe, they'll move to the phase 0. Yes, Sharon.
Yes, sir. Phase 0 of a clinical trial is done with a very small number of people, usually fewer than 15. Investigators use a very small dose of medication to make sure it is not harmful to humans before they start using it in higher doses for later phases. If the medication acts differently than expected, the investigators will likely to do some additional preclinical research before deciding whether to continue the trial. Yes, sir.
Okay. Now everything is fine, guys. In the right hand side, you can see the advertisement. That is crazy, right? Okay, now let's come back to this. What happens in phase 0? Phase 0 of clinical trial is done with a very small number of people, usually fewer than 15. Investigators use a very small dose of medication to make sure it isn't harmful to humans before they start using it in higher doses for later phases. So what they're trying to say here, in phase 0, a very small number of people will be enrolled, normally fewer than 15. Investigators, doctor use a very small dose of medication. There are doses of medication, 5 mg, 10 mg, 15 mg, 100 mg. So they use a very small dose of medication to make sure it isn't harmful to humans. To make sure the drug is not harmful to humans, because if they initially give the higher dose, it can have a side effect. Okay. If the medication acts differently than expected, then investigator will likely to do some additional preclinical research before deciding whether to continue the trial. Okay. Now let's move to phase 1. Yes, Sharon.
During phase 1 of a clinical trial, investigators spend several months looking at the effects of the medication on about 20 to 80 people who have no underlying health conditions. This phase aims to figure out the highest dose humans can take without serious side effects. Investigators monitor participants very closely to see how their bodies react to the medication during this phase. While preclinical research usually provides some general information about dosing, the effects of a medication on the human body can be unpredictable. In addition to evaluating safety and ideal dosage, investigators also look at the best way to administer the drug, such as orally, intravenously, or topically. According to FDA, approximately 70% of medications move on to phase 2.
Okay. So there's in phase 1. During the phase 1 of clinical trial, investigators spend several months looking at the effects of the medication on about 20 to 80 people. So you can see here a difference. On phase 0, there were less than 15 people. In phase 1, 20 to 80 people. In phase 2, several hundred participants. So as we are moving to, the next phase, the sample size is getting increased, or we are increasing the sample size. This is common. And the months, the duration of, investigation is also increasing. So with every increasing phases, we are having around 4 phases. The duration and the sample size, means the number of participant gonna increase. So here you can see, here we are selecting the people who have no underlying health condition. What does it mean? Healthy people. They are selecting healthy people. So why they are not selecting patient or the people with the disease?
Because they will be already under different medications. So there might be some effect of that, both the medications, and there
Correct.
we cannot analyze the condition also.
Correct. But
Side effects we cannot analyze.
correct. But Malti, let's suppose we are making a medication for cancer. Until unless we got an opportunity to test on cancer patient, how it gonna be decided that the medication is working correctly or not? So what happened in the initial phase? The phase 1 is an initial phase. So initially they're gonna test on healthy people because healthy people immune system will be very good. So they can bear the things if any side effects are there. If in the initial phase they're gonna enroll patient with the disease, so there can be side effects because already they are suffering from the disease. And if the drug is not proper, it can have a lot of side effect. It can harm their health, but it can cause death as well. That's why in the initial phase, like phase 0 or phase 1, we're gonna take healthy people, people with no underlying health condition. This phase aims to figure out the highest dose human can take without serious side effect. See, what is the main purpose of this phase? The phase aims to figure out the highest dose human can take without serious side effect. It means, according to the severity of your disease, doctor suggests the dosage level, right? Sometimes to some patient, they give 5 mg, to some patient 10 mg, some patient 100 mg or 200 mg. So how they decided or how they decide this is safe because they are testing on which phase? Phase 1. So in this phase, they'll give the patient the highest dose human can take without serious side effect. First, they'll give 5 mg, maybe whatever they think, the smallest dose, then 10 mg or 100 mg. And they'll check how their bodies are reacting. And they'll stop going beyond once the bodies of this patient or volunteers are having side effect. That's how they test. Okay. Now, these are common things which is written while preclinical research usually proves general information. So this is not needed much. So every phase will have few things. First, the duration of the phase, participants or sample size, and then the aim of the phase, what they want to achieve or what they are trying to achieve from this particular phase. So these three things you need to always remember. How much is the duration? How much will be the sample size? And what's the aim of the phase? And another part is like in that particular phase, healthy people are there or people with the disease. Okay. Now let's move to phase 2. Yes, Sharon.
Phase 2 of a clinical trial involves several hundred participants who are living with the condition that the new medication is meant to treat. They are usually given the same dose that was found to be safe in the previous phase. Investigators monitor participants for several months or years to see how effective the medication is and to gather more information about any side effects it might cause. While phase 2 involves more participants than earlier phases, it's still not large enough to demonstrate the overall safety of a medication. However, the data collected during this phase helps investigators come up with methods for conducting phase 3. The FDA estimates that about 33% of medications move on to phase 3.
Now what happens in phase 2? Phase 2 of a clinical trial involves several hundred participants who are living with the condition that the new medication is meant to treat. So you can see here we are taking patient, people with the disease. They are usually given the same dose that was found to be safe in the previous phase, because in the phase 1 we have tested, right, the highest dose human can take. So the same level of dosage will be given here. They'll not increase the dosage here. Investigator monitor participant for several months or years to see how effective the medication is, to gather more information about any side effect it might cause. Okay, so what they are doing, they are investigating the participant for several months or years to see how effective the medication is and to gather more information. Okay, and now this is common information, like while phase 2 involves more participant than earlier phases, it is still not large enough to demonstrate. Okay, now what is something which is very new in this phase? This is the first time they are taking people who are living with the condition that the new medication is meant to treat. And they are observing the changes or the reactions on their body, how their bodies are reacting. That is written in this para, second one. Now these are common information, like FDA estimate 33% of medication move on phase 3. Now let's see phase 3. Yes, Sharon.
Phase 3 of a clinical study usually involves up to 3,000 participants who have the condition that the new medication is meant to treat. Trials in this phase can last for several years. The purpose of phase 3 is to evaluate how the new medication works in comparison to existing medications for the same condition. To move forward with the trial, investigators need to demonstrate that the medication is at least as safe and effective as existing treatment options. To do this, investigators use a process called randomization. This involves randomly choosing some participants to receive the new medication and others to receive an existing medication. Phase 3 trials are usually double-blind, which means that neither the participant nor the investigator knows which medication the participant is taking. This helps to eliminate bias when interpreting results. The FDA usually requires a phase 3 clinical trial before approving a new medication. Due to the larger number of participants and longer duration or phase 3, rare and long-term side effects are more likely to show up during this phase.
Okay. Now let's see what happens in phase 3. Phase 3 trials are usually involve up to 3,000 participants who have the condition that the new medication is meant to treat. So you can see from phase 2, they are taking patient from phase 2. Before phase 2, they were taking healthy people. In phase 2 also, they are taking patient, and phase 3 also. And here you can see, as I have told, with every increasing number of phases, they are increasing the number of participants. So here more than 3 or up to 3,000 participants. Okay. The purpose of this phase 3 is to evaluate how new medication works in comparison to existing medication for the same condition. To move forward with the trial, investigators need to demonstrate that the new medication is at least safe and effective than the existing. Now what they have written, this is not the exactly same. Why? Because here they have written the purpose of phase 3 is to evaluate how new medication works in comparison to existing medication. But this is not the case. Every time it is not mandatory that there is an existing medication. Let's suppose in the COVID scenario, no drug was there. So how they can compare? There is no same medication for the COVID. So normally what happens, let's suppose if already, for the same treatment, there are multiple companies who create drugs, right? So if there is already a medication which is available for the same treatment, so the next company who is creating the medication, they have to prove that their medication is at least safe and effective compared to existing medication. Because if it is not same and effective, what's the reason FDA will approve? Or what's the reason of existence? If already a medication is available and the new one is not better or still same than that. But let's suppose there can be a scenario that there will be no medication for comparison. Okay, so if there is no medication for comparison, then this will be null and void, will not compare. So what will happen in the phase 3? To do this, investigator use a process called randomization. This involves randomly choosing some participant to receive the new medication and other to receive existing medication. Okay. Let's suppose if 20 people are there, they will divide into 2 groups. 10 people will receive placebo, sugar pill or dummy drug. 10 people will receive active drug, which we are creating. Why they do this? They want to compare the result. Sometimes what happen, people believe that they are taking the drug and they heal by their own. So they compare, is they are healing by their own or by our product, the active drug. So they'll compare the result of these two groups. Okay. Now how they do? The phase 3 trials usually double blinded. What does it mean by double blinded? Neither the investigator, which means neither the participant nor the investigator know which medication the participant is taking. It means the drug we are giving to the patient, doctor doesn't know which drug he is giving to which patient. Why it is done? This helps to eliminate the bias when interpreting result. Let's suppose I am a doctor. So there are some participants, but some participants are from my village or in my relations. So what I'll do, I'll try to avoid giving them the active drug which can harm their body. I'll give them placebo, dummy drug, or already a proven drug, which is better. But to eliminate this, they do this double blinded. So doctor doesn't know which drug they are giving to which patient. Everything is coded with numbers. Okay, the FDA usually requires phase 3 clinical trial before approving new medication due to the large number of participants and longer duration. Okay, these are external things. If you want, you can read, but till here it was important part. They have written the numbers as well, how many studies move to next phase. Roughly 25 to 30% of medication move to phase 4. Okay, now what happens in phase 4? Yes, Sharon.
Phase 4 clinical trials happen after the FDA has approved medication. This phase involves thousands of participants and can last for many years. Investigators use this phase to get more information about the medication's long-term safety, effectiveness, and any other benefits.
Okay, so one thing is that in phase 3, if investigators demonstrate that the medication is at least safe and effective as other already on market, the FDA will usually approve the medication. After the phase 3 or in the phase 3, if investigators are able to demonstrate that their drug is safe and effective, then it will be approved. So if after the phase 3 it will be approved, then what's the reason we need phase 4? The phase 4 clinical trial happens after FDA has approved medication. It means if phase 4 is happening for that drug, it means it's already approved. It's available in the market. This phase involves thousands of participants and can last for several or many years. Investigators use this phase to get more information about medication, long-term safety, effectiveness, and any other benefit. So, once doctor suggests drug to patient, so they'll tell you like, take this drug and you can again come back after 4 days or 10 days. And if you're feeling not well or anything is there, they'll schedule some, visits. So the patient will come back and they'll tell, yeah, there is an improvement or I'm feeling itches or any headache or anything. So those feedbacks will be collected by the doctor. And again, this feedback will go back to the company. So this is long time. It can last for many years. It can be 20 years, 30 years. Hey, what's the reason of phase 4? If till phase 3 we have done a lot of inspection. So there was a case in US. There was a lady, she was using Johnson & Johnson's baby powder. And due to that, she have to suffer with breast cancer. So then she filed a complaint for Johnson & Johnson's, and Johnson & Johnson have to pay, huge amount of money, and it impacted on their brand name as well. So that's how it is very important. What is the long-term safety or efficacy of the drug, any product? No, you can say Johnson & Johnson, like why they were sued for a baby powder. So see, baby powder is also a product. No one should get an cancer, right? So any product you are also, it's not only the drug on which clinical trials are happen. It's on cosmetics as well. Any cosmetics you are applying on your body, if it is the brand. So, and you know, Johnson & Johnson, a lot of people know them with baby products, but they are one of the top CROs in the world. Okay. So that's how this phase 4, because doctors will continuously collect the data maybe for 10, 20, 30, 40 years. So they can improve their drug. They can make some changes. So this was about phase 4. Now what you guys need to do, you need to create an PPT. You all need to create one PPT from your end. And you just need to keep it simple and some points only. And few things you need to remember, like what is the sample size of the particular phase? Duration. Sample size means how many participants are there and what is the duration? Then are they healthy people? Are they patient? Then what is the AM of that phase? 3 or 4 things are there. So 3 or 4 points in each phases are enough. Any questions? Anyone for now?
Sir, I have a doubt, sir.
Mhm.
Sir, in this process of randomization, so this is in the phase 3, right, sir? Some 2,000 to 3,000 people are participating, right? 3,000 participants. So in this randomization process, all the 3,000 patients are grouped into different groups, or only they will randomly pick the people?
Randomly.
Randomly, they will pick either of, it might be 1,000 people or 500 people, they will pick out of these 3,000 people.
Yeah, they'll divide into groups. It depend upon them. Like they'll, let's suppose 3,000, so maybe 1,500. So if it is written 3,000, it doesn't mean that mandatorily they have to take 3,000. It depend upon the study, company and the team, like what they are deciding. If they want to take 1,000 or 500, they can also take.
Okay. Okay, then they are going to test this by making groups on all the participants who are there in the phase 3. Or it is like, suppose say 1,500 are participating in a phase 3 trial. So how many they are going to pick? All the 1,500 will participate?
They'll divide into 2 groups.
Okay, okay,
Half
got
half.
it, sir.
For
Okay,
half
got it.
of them, they'll give the placebo or dummy drug, sugar pill.
Okay.
Half of them, they might give the active drug. Or if there is an existing medication, so they'll give the existing medication for half of them, and for half of them, the new one, active drug.
Okay. Okay, then what is the use of giving that dummy drug or placebo, sir?
Because I told now that a lot of people, they feel that I am taking the active drug and they heal. This happened in the past. Sometimes people, they think that I'm taking, because they don't know, right? The patient doesn't know which drug they are taking. So sometimes they feel that I'm taking the drug and they heal. So we are interpreting the result whether they are healing by their self or with our drug. So we'll compare the results, the placebo drug results with the active drug results. Because placebo is nothing but a sugar pill, right? If they are healing and the active drug, the people who are consuming active drug, they are not healing, it means our drug is not useful.
Okay. Okay, sir. But why they want to compare with the normal? Because this is like for particular condition only we are giving a pill, right, sir? Why we wanted to test for the null
Yeah.
condition?
There are 2 reasons. Let's suppose if there is an existing medication, so they get a comparison, right? How it is performing compared to existing medication, right?
Yes, sir.
So they can prove it is better, or at least in comparison with them, it is relevant, right? At least safe and effective. That is one thing. Another, that to prove that if there is no existing medication, they'll give the placebo dummy drug, and they'll compare with that people are not healing by themselves, they are healing by our drug. You can prove, right? There is a reference.
Got it.
We'll have the data of people who are taking dummy drug, that is nothing but a sugar pill, and people who are taking the active drugs. So you can prove these are two cohorts, and the people who took our drug, they have healed, or we are able to treat their disease.
Got
Right.
it, sir. Got it, sir. Got it. Thank you, sir.
So if there is no cold, will not understand the difference between heat and cold. If every time heat is there, so if cold is there, we understand, okay, this is like hot or this is cold. So that's how here also, like, they prove it with nothing. Patient who are receiving nothing, a sugar pill, with the patient who are receiving the new drug, active drug, and that's how they interpret it. Just read it, guys. Just spend some time on this, create an PPT, and that's how you'll get more understanding. Any questions, anyone, for now? Yes, Sharon.
Sir, I am sharing my screen.
Yeah.
Sir, now can you see my screen?
Yeah.
Sir, here row number 143. Sir, what is this, sir? Put total number of enrolled patients. I didn't understand this line.
What is the question here?
Here we are applying do while to enroll the patients. To enroll at least 100 patients.
The question is, suppose we are conducting a clinical trial and we want to enroll patient. Until we reach to total enrollment at least 100 participant, what condition loop you will use? Because both do until and do while will draw the same result. And here we are using do while. Okay. Total number of enrolled patient. So, okay, what was your question? Why we have written
this line, sir? Put total number of enrolled patients and colon this.
Okay, just run this.
So without this also we are getting the results, sir. Without this line also.
This is something looking like a title. Just go in the output data. Click on this variable name total enrollment. Here, yeah. Go back to coding window.
Sir, maybe put is used for output data in a log window.
It seems like maybe. Okay, go in the log. Yeah, that's correct. Here you can see total number of enrolled patients are 100. That's correct.
I didn't understand, sir.
So you are getting this line right before note, total number of enrolled patient and total enrollment is 100. Can you see this?
Yes, yes.
This you are getting because we have written that. So this
Okay.
after put we have written this total number of
Total
enrolled
number of
patient, right?
patients.
And then we have given the variable name total enrollment, right? Because of that variable, it is taking the data from a variable.
Okay.
It is simply putting the result along with the note you have written in the log window.
Okay. Okay, sir. And
Sir, is it not visible in the output, sir?
Sorry.
Is it not visible in the output?
Just go in the output data window. It's visible 100.
no, sir, she is asking, I think total number of enrolled patients.
Total
This
number of
it is
enrolled
not
patients.
visible in.
No, that is visible that we have written for the log. So it will
Okay.
not come in the data.
Okay, sir.
What is meant by writing for the log, sir?
What's like you are asking, Malti, what's the meaning of writing in the log?
Yes, sir.
Simply to see if you don't want to open the data set, if you want the some information to be present in the log, so you can put it or you can show there.
Okay. Put function we can use for this also.
Yes.
We used to
Yeah.
convert character variables and numeric variables into character variables, right, sir? Apart from that, we can use this also. Like,
No, that is separate. See. See, you remember we have learned input statement and then input as in converting variable type. So you can
yes,
see at one point of time input is used to create variables. At one point of time input is used to convert character variable to numeric. Same way, at one point of time put is used to convert numeric to character, and here put is used to display. So it depends, Malti, in which scenario you are using that particular statement.
okay.
If you are writing input after data statement, like after data name, you are writing input in the next row, it means you want to create variables. If you are writing input as a function, right? Input as a function, if you are writing, it means you want to convert it, right? You want to convert. But if you are writing put as a statement, here it is as a statement, right? Put. If you are writing put separately as a statement, it means you want to display something.
Okay. Here put will display something only in the log window.
Correct.
Okay. And generally, in what scenarios we generally use this kind of?
It's very simple. See, if you are using put as in function, what will be the scenario? You want to convert the variable, right? Variable
Yes.
type. If you are using put here separately as a statement, it means you want to display something in the log window.
Okay. How it is applicable for the while working? I'm just asking in what scenarios we will.
That's very simple. Let's suppose there is something from the data you want to display in the log. So you can write in this scenario whatever note you want to write, and you can give the variable name. It will display that particular data in the log. Like you can see total number of enrollment patient, and you can see 100. Total enroll is equal to 100. So this information, how many total enrollment is there in your data from your data? So that is showing in the log window.
Okay, sir.
Okay.
Yes, sir.
Sir,
Sir, I asked you about the log errors also. Like
Sorry.
teachers log errors. Like it is writing like 322 to 322.
That is something pending. Like few things I told I'll be covering. What was those things. Like one was log errors. I told I'll be creating some data set so that I can show you. One was log error and another was anyone? Okay. So Malti, do you have that code? You had a code, right, where you are getting that log error.
Sir, exactly now I don't remember because it was
No
somewhere
worries,
in
no
a
worries.
book.
Yeah, I'll create it, no worries.
Sir, Sharun was asking something. I think I have
Yeah,
interrupted.
yes, Sharon. Yeah.
Sir, here, sir, in this row, 223 row, so we are writing all this adds the observation from one source data set to, we are writing all this and we have not commented it, but also it
It's
is
working,
showing the
right?
correct result. Yes.
Yeah. Actually, there are multiple ways to comment, Sharon. One way is on 228 row. That in the beginning and end, this forward slash and asterisk symbol should be there. Okay, one way is that. Another way is like if you want to comment inside a data, let's suppose in the beginning proc append is there and at the end run is there, and beside them usually what should be there? Codes, right?
Yes.
So but here isn't comment. So if you want to comment inside the code, so what you can do, in the beginning you can give asterisk symbol and semicolon, because whenever we end a statement, right, code statement, we give semicolon. So just you mention beginning, asterisk symbol and semicolon, it will get commented automatically. But this will work mostly if you are commenting inside the code, not separately.
Okay.
Like you can see 221 row, that is separate. So you have given forward slash and asterisk. But inside
Yes.
this, you have used different method, asterisk and comma.
Okay. Okay, sir. And, oh, sir, here, sir, here underscore type underscore and underscore freak underscore are the default variables. So freak
Yeah,
means it is frequency, choose the counter. So what is this type, sir?
this is also default. First, it is showing for total.
Yeah.
00 is for total, and then 11 is for female and male.
Yes. Sir, here 19 means we are having total 19 observations, and 9 means we are having total 9 observations with female F. So what does it mean 0, 1? What does it mean, sir?
This is not needed. You can just simply neglect. These are default variables. It will automatically every time generate. The end goal is to get the statistics for the particular variable.
Okay. Sir, and in proc transpose and proc rank, proc append, and proc summary, we need to write proc print. While in other proc steps, we don't need to write proc print. So why is that, sir?
See, in uh, proc means if you are selecting any variable and everyone already know that what is the use of proc means to generate the statistics. So what proc means will do, it will automatically print those statistics in the result window. But in proc transpose, that is totally different thing. You decide that which the output is not default. Here the output is default, Sharon. In proc means every time you select a variable, the output gonna be same, right? Until unless you are manually changing it. But proc transpose output not gonna be same, right? So that's why it gives you the control that whether you want to print or not.
Okay.
But here it will by default, like proc means proc freak, by default it will get printed, right? Output is default, fixed. It will directly show you in the result window. And most probably you want to see the statistics.
Yes.
Is the value is correct or not?
Okay. Okay, sir. That's it.
So guys, for now, create a PPT on clinical trial phases. I'm sharing you the link in the group itself. You can refer this website. Let me share it right away. I have shared in the group. Okay, so guys, let's meet in the tomorrow session. And just allow me for a minute. Okay. So